Electrophoresis. Okay. Today we discuss about interesting topic electrophoresis. What is mean by electrophoresis? The movement of charged particles in an electric field resulting in their migration towards the oppositely charged electrode is known as electrophoresis. Okay, that is called electrophoresis. Look at here. Next, definition of electrophoresis. Electrophoresis is a laboratory technique used to separate DNA, RNA, or protein molecules based on their size and electrical charge. Okay, electrical charge means positive and negative charges. Okay, and electric current is used to move molecules to be separated through a gel. Okay, that is called electrophoresis. Okay, next, Arne Tislius in 1931, while new separation process and chemical analysis techniques based on electrophoresis continue to be developed in the 21 21st century. Okay, simply I say what is meant by electrophoresis. Okay, we have to supply the power into both the trace. Okay, positive and negative charges. Okay, the technique for the separation of biological molecules such as plasma proteins, lipoproteins, and immunoglobulins. Okay, we have current supply. Okay, separation of biomolecules. Okay, that is called electrophoresis. Electrophoresis is a widely used analytical technique for separation of biological molecules. Okay. Next, types of electrophoresis. Okay. First one is zone electrophoresis. Next, second one is immunoelectrophoresis. Third one, isoelectric focusing. First, zone electrophoresis. Zone electrophoresis, there are two types. First one is paper electrophoresis. Next, gel electrophoresis. Okay, look at here. Zone electrophoresis, a simple and modified method of moving electrophoresis in the zone electrophoresis. Okay, and inert supporting material such as paper or gel are used. Okay, that is called zone electrophoresis. Okay, first one is paper electrophoresis. It is a technique that use Watsman number one filter paper. It can be used. Okay, look at here. This is paper electrophoresis experiment. Okay, first you take Watsman number one filter paper. This is Watsman number one filter paper. Okay, first you take capillary tube. With the help of capillary tube, we have to take a sample as a drop on the paper. Look at here. This is the drop on the paper. This is Watsman number one filter paper. Okay, we have take two trays and pour the buffer solution. Look at here. This is two trays. Okay, this is buffer solution, blue color. Look at here. This is buffer solution. Okay, as shown in the figure A, we have to dip the paper in a buffer solution. Okay, we have to dip the paper in the buffer solution. Okay. Next, we have to supply the power into both trays, positive and negative charges. Okay. What happened? Okay. Molecules with a net positive charge move positive charge to move towards the negative cathodes. Okay. While those with negative charge migrate towards positive anode. Okay. Next, with the effect of electric supply, you can observe the following changes in this experiment. Okay, colors will be formed. Okay, that is called biomolecules. Biomolecules based on color observation. These might be proteins. Okay, or amino acids. 1 minute. Look at here. First step, it is a technique that use Watson filter number one paper. Okay, moistened by a buffer and then connected by two ions electrode. Look at here. Okay, this is positive, this is negative electrodes. Next step, then the sample is applied on one and left for separation of the component under electric field. Okay, that is called positive and negative charges. After separation, the filter paper is dried and strained to get colored bands. Look at here. This is color bands. Okay. Total 1, 2, 3, 4, 5, 6, 7. 7 bands. Okay. This is negative ions. This is positive ions. Okay. This is the sample. Look at here. This is sample. Okay. Point of sample application. This is negative ions. This is positive ions. Look at here. After separation, the filter paper is dried and strained to get colored bands. These colored bands are recognized for the natural sample by comparing with the standard. For a sample of sterol, 5 bands of protein can be separated. Okay, that is alpha 1 to beta. Next one, gamma. Okay, fifth one, albumin. Okay, total 5 samples. This is example of paper electrophoresis. Okay, next, paper electrophoresis has been extensively used in almost all laboratories where proteins and other similar macromolecular electrolytes are investigated. Next, excellent sharply defined separation of serum proteins. Paper electrophoresis is still of particular interest where small amount of protein needed to be isolated for further analysis or testings. Okay, this is paper electrophoresis. Okay, next, albumin. Look at here. This is egg protein. Okay, this is example of one sample. Okay, any doubts? Once again, I repeat. Look at here. First, definition of electrophoresis. Electrophoresis is a laboratory technique used to separate DNA, RNA, or protein molecules based on their size and electrical charge. Okay. Electric current is used to move molecules to be separated of biomolecules. Okay, separation of biological molecules such as plasma proteins, lipoproteins, and immunoglobulins. RNA This first invented by electrophoresis technique. Okay, next types of electrophoresis. First one is zone electrophoresis. Second one immunoelectrophoresis. Third one isoelectric focusing. Okay. First one is zone electrophoresis. Zone electrophoresis means it can be used two types of material. It can be used. Okay, that is called paper or gel. Okay, that is called zone electrophoresis. Next one, first one is paper electrophoresis. In this technique, it can be used very important. Watsman number one filter paper, it can be used. Okay, next, look at here. In this technique, the sample is applied on a strip of filter paper wetted with desired buffer solution. Look at here, this is buffer solution. The ends of the strip are dipped into the buffer reservoirs. Okay. Next, which the electrodes are placed, the electric current is applied, allowing the molecules to migrate for sufficient time. The paper is removed, dried, and strained with a dye that specifically colors the substances to be detected. Look at here. This is spot. Okay. The colored spot can be identified by comparing with a set of standard run simultaneously. Okay. For the separation of serum proteins, Wattsman number one filter paper, uh, it can be used. The buffer pH 8.6, it is maintained. Okay. Okay. This is examples of electrophoresis sample. Okay. Alpha 1 to beta gamma albumin. Okay. Total 5 samples. Okay. Next, any doubts? This is paper electrophoresis. Okay.